Hello, everyone. My name is Emily Hill, and I am currently a senior in the Department of Ecology and Evolution. And this is my poster for the 2020 Undergraduate Symposium. So first, a little introduction into my research. My lab is actively studying the ecology of microbes, specifically in the soil. Soil microbes are ubiquitous throughout the environment and play very important roles in the productivity and longevity of both soil and plant communities. For example, they are heavily involved in nutrient cycling in the soil, as well as forming associations with plants. Plant-associated microbes exist along a mutualistic to parasitic spectrum and can also heavily influence the environment around them. So we wanted to study these plant-associated microbes, and we study them specifically in agricultural systems, largely in part because human land use in the terms of agriculture has been shown to lead to changes in plant community composition as well as soil degradation, but there is still little is known about the underground community effect on agricultural management. So for this research, we use vineyards as our model system due to the various management practices employed, so we can compare those, and as well as grapevine plants have been shown to harbor many mutualistic fungi. For this poster, these are arbustial mycorrhizal fungi, and then as well as host a variety of pathogens. So my research questions are, how do agricultural management practices such as tillage and nutrient amendments affect the composition of these fungal communities? And do different functional groups, fungi, for example, mutualists and pathogens, respond to the management in the same way? So these are what I kind of want to look at. So with this study, we conducted this study. We first collected soil cores from 76 vineyards across Oregon in the summer of 2018. These soil cores were collected right near the vine. So in this picture with the little diamonds, that's where we took them. Fungal DNA was then extracted and sequenced to derive our fungal mutualists and our fungal pathogens. In figure 1, we found that there was a significant change in community composition in both fungi and pathogens in tillage practices. So we see different species in no-till and till soils. In figure 2, we found that fungi diversity was altered by management practices. We saw that there's increased diversity in more organic fertilizers compared to more conventional mineral fertilizers. However, we did not find any significant diversity changes in pathogens, and we also didn't find significant diversity changes in till soils in both fungi and pathogens. So compared to our top figure, we saw a different species composition, but we actually don't see a change in the number of species. And then in figure 3, we looked at fungal community composition in the management, and I split them into different precipitation gradients, as there is some research showing that fungal communities can be structured differently due to different environmental patterns. So one of these being precipitation. So what we see here is that fungal communities composition is affected by management, and we also see that they cluster differently between precipitation. So we don't see the dots being in the same shape or form across precipitation. So from these results, we found that both fungi and pathogen community composition was heavily influenced by agricultural fertility management. So the species we're seeing are very different in the different managements. However, we did not find significant diversity changes, only with fungi in management, but with all the other three, we did not. So from this, these results kind of highlight the importance of agricultural management practices on the soil microbiome, as well as kind of the need to evaluate the effects of fertilization treatments on communities for both pathogens and mutualists, because they are so important for our environment, and we really need to provide soil with these necessary functions that these microbes give off. So yeah, that's my poster. Thank you for listening.